In this part of the lesson series on neurons, we'll take a look at how a neuron generates its output signal, called an action potential, how that electrical signal races down the axon to the terminal. Now, we've learned before that the electrical nature of neurons involves charged particles moving into and out of the cell. So whenever charged particles are moving, that's an electrical current. The cell membrane is the boundary of the cell, but within the membrane are these protein channels. These are molecules that can act like little channels to allow ions to move across the membrane, and that constitutes an electrical current. The action potential is generated at the base of the cell body, so where the axon meets the cell body. So it starts right at that location. Now, if we could zoom into the surface of the axon membrane, we would see that it is studded with two different kinds of protein channels. Those protein channels, the red ones here, we're going to call them the sodium channels, and the blue ones are potassium channels. Sodium is a charged ion, part of sodium chloride, table salt, and potassium is a charged ion. They both happen to have positive charges. Now, as it happens, there's more sodium outside the cell, in the fluid outside the cell, and there's more potassium inside the cell. So we have this distribution of ions that is key to how neurons operate. Now, what's going to happen then? The beginning of the action potential happens when these first sodium channels open up. And we'll describe in a minute how do these channels know to open up. But when these sodium channels open up, the sodium outside the cell can rush in through those channels. And it's a combination of electrical forces that move the sodium in, as well as concentration differences. There's more sodium outside than inside. They tend to move into the cell. And when sodium rushes into the cell, the effect of that is to trigger the opening of the potassium channels. Well, now potassium can leave the cell, again, due to electrical and concentration factors. And so what we have then is we have a sequential opening of these channels in the membrane of the axon, allowing sodium ions in and potassium ions out. So it's sort of like a domino effect. So these channels open, then these channels open, then these channels open, then these channels open, and ions are going in and out. This is the nature of the electrical disturbance that we call the action potential. It's an electrical signal. It's a wave of channel openings with local movement of charged particles, these sodium and potassium ions. Now let's try to understand what is it that triggers the first set of sodium channels here that will then lead to the movement of the action potential all the way down the axon. Well, these channels open up when enough positively charged ions, enough sodium, has entered the cell from synapses all around the cell body and on the dendrites. Remember, at each synapse, there can be a local electrical signal being generated. In this case, we see transmitters released, binding to the receptor, the channel opens, and in comes sodium. Well, if enough sodium enters the target cell, along the dendrites and in the cell body, if enough gets in, we call that the threshold. If enough sodium gets in, it will trigger the opening of these channels. Notice then, these channels on the axon are not responding to chemicals like at the synapse. These channels are sensitive to the charge inside the cell. And when synapses are active and sodium is rushing in, then that drives the cell past this threshold amount of electrical current, and these channels are sensitive to that charge entering the cell. They open up, and then you get that wave of activity that we call the action potential. Now let's take a look at how the output signal is reflecting the activity of synapses that make contact with the target cell. So here's a target cell, and here's the communicating neuron. If the communicating neuron releases just a small quantity of neurotransmitters, because maybe only one electrical signal raced down to the terminal, so we get a little bit of neurotransmitter out, well, those neurotransmitters will bind to receptors, but it'll be a small electrical current here. And it may not reach threshold, so the target cell does not even, there's no electrical signal, doesn't even respond. But let's suppose the communicating neuron has a higher level of activity. So multiple electrical signals race down to the terminal. Well, more neurotransmitters are released. With more neurotransmitters, it's like having more keys to open more locks. The receptors here, the channels open up, more ions rush through. You get a bigger electrical signal in the target cell, and you might pass threshold whereby the target cell generates 2 action potentials in response to the increased activity here. If there's high frequency electrical signals racing down to the terminal, we get lots of neurotransmitter released, which means we have a larger electrical current, and that means you go way past the threshold, and the target cell responds with high frequency action potentials as a result. So notice then, the target cell is responding in a graded fashion. When there's not a lot of synaptic activity, it may not respond at all. When there's a little bit, it responds with low frequency electrical signals. And when there's a lot of activity at the synapse, it responds with high frequency signals. Now, we said earlier that the cell body is integrating incoming signals and then generates the outgoing signal. Well, what's the nature of this integration? Well, keep in mind there may be hundreds or thousands of synapses all over the dendrites in the cell body. So the target cell here is going to, in a sense, integrate all of the electrical currents that are being generated at active synapses. And now we have to introduce another kind of synapse. We've already seen that a synapse can be excitatory. It can excite the target cell. So the neurotransmitter opens up channels that allow positively charged particles like sodium into the cell. That will tend to make the cell respond with an action potential. But there are other synapses that have the opposite effect. They block the target cell. They prevent the target cell from generating an electrical signal. We call this inhibition. And it has to do with the nature of the chemical being released, the neurotransmitter here, and its corresponding receptor. So if it's an inhibitory synapse, then what happens is these channels are being opened up by the neurotransmitter, but it's not sodium that's rushing it, it's chloride ions, and chloride ions are negatively charged. So it can, in a sense, counteract the positively charged sodium ions that are rushing in. So it can cancel out the effect of excitation at other synapses. So what we have then, we have a situation where we have a neuron that may have thousands of synapses, some of which, like in green, will tend to excite the target cell, but some of which, in red, are inhibitory. And so it's the overall balance of activity of excitation and inhibition that determines whether or not the target cell will respond. Down here we see an electron microscope picture. This is a scanning electron microscope picture showing a bunch of synapses on the cell body of a target cell. In summary then, the neuron can generate an output signal. It begins at the base of the axon, and it's carried by a wave of channel openings that allow sodium ions in and potassium ions out, and that disturbance goes all the way down to the axon terminal. These first sodium channels open up when a threshold amount of sodium ions enter the target cell. If sodium's coming in at a synapse location, that's an excitatory synapse. It's exciting the target cell. However, other synapses can allow negatively charged ions into the target cell. Those would be inhibitory synapses. Negatively charged ions like chloride would tend to cancel out the influx of sodium. So it's the balance of activity, the balance of excitation and inhibition that determines whether these sodium channels on the axon are going to generate the action potential as an output signal. So the integration amounts to a kind of metaphorical decision. The target cell is deciding whether or not to generate its own electrical signal. So here we see then how neurons are chemically and electrically excitable. Chemicals released from axon terminals can open up ion channels. If positively charged ions like sodium enter, that will tend to excite the target cell. If negatively charged ions enter, that will tend to inhibit the target cell. But if enough positively charged ions get in to reach threshold, then those first set of sodium channels on the base of the axon open up. That triggers the neighboring potassium channels and the neighboring sodium channels, and you get that wave of channel openings, and the action potential races down the axon to the terminal. And then, of course, chemicals are released from the terminal onto the next cell in the chain.